waxy layer called the cuticle. And that's a problem for photosynthesis, because if you cover yourself with wax, then you can't carry out gas exchange, which means CO2 can't get into the leaf. Now, the reason that the plants cover themselves with wax is because most of the volume of the cells is water. And if you're exposed to the dry air outside, what happens to your water? Evaporates. So you can't really afford to lose a lot of water, but you need to be able to get CO2 to photosynthesize, which means you have to expose yourself to the air, but if you expose yourself to the air too much, you dry out. And water is usually the limiting factor where plants are growing, unless if it's an aquatic plant. So you cover yourself with wax to prevent water loss, but that creates a problem because now you can't get CO2. And you need photosynthesize, but you can't afford to lose too much water. Yes, sir? You bring me the next point. So they do have the waxy layer over their skin layer, but they provide these little pores in the skin called stomata. And when the pores open, CO2 can go in and water can come out. You don't want the water to come out, but there's no choice. And when the stomata close, you don't get any gas exchange. So these two slides we made in the lab. The bottom one is actually an epidermal peel. So we took some Zebrina plant, and the common name for this, I'm sure you've seen this plant, and I should bring you a picture at some point, has these nice green and silver stripes on the top of the leaf, and it's this color on the bottom of the leaf. So some of you have seen those as house plants. If you've gone to any nurseries to buy them, the common English name, and I have no idea what that's the common English name, but the common English name is the wandering Jew. It's a creeper, so I don't know why it's called that. It doesn't grow in the desert or anything. But so in nurseries, they call it that, as opposed to Zebrina, but the genus name is Zebrina. Here's the pore. These are the epidermal cells. The epidermal cells are tightly packed, and they're covered with a waxy cuticle, so they don't let gas exchange to happen. But here's a pore, and the pore is guarded by two cells. These two cells are called guard cells. So when the pore is open, you see the little slit here between the two cells, and when the two cells collapse, the pore closes. And the guard cells do have chloroplasts in them. So you see these little green dots, those are chloroplasts. The rest of the epidermis does not have any chloroplasts, because the role of the epidermis is protection. But the guard cells open and close the stoma, which allows for gas exchange. The rest of the epidermis is covered in the wax. The top slide is not an epidermal peel. This is a cast. So the way we make this is you paint your nail polish on the leaf, let it dry, peel it off, look at it under a microscope. So you can see the epidermal cells, and you can see the stoma with the two guard cells. Here's another stoma with the two guard cells. Here's another stoma with the two guard cells. So we do this so that we can look at the density of the stomata, the size of the stomata, things like that. So far, so good? All right. Well, not all leaves have stomata at the same place. So here is a picture of a privet plant. Privet has pretty much horizontally oriented leaves. Here's a cross-section of a leaf from a privet plant. It has an epidermis, the skin layer at the top. It has an epidermis skin layer at the bottom. There's a cuticle, the waxy layer covering the epidermis at the top. There's cuticle, the waxy layer covering the epidermis at the bottom. There are two types of photosynthetic cells. The photosynthetic cells are called mesophyll cells, and there's the palisade and the spongy. The palisade mesophyll cells are column-shaped. They are tightly packed. Lots and lots of chloroplasts that are stained purple. The spongy cells are smaller, lots and lots of air spaces in between them, and they have fewer chloroplasts. Notice there are stomata in the bottom side, but there are no stomata at the top layer. So the upper epidermis does not have any stomata. The lower epidermis does have stomata. So if you think of how the shape and anatomy of this leaf would relate to its function as a horizontally oriented leaf. Where's the light coming from? Always at the top. Whether it's morning, afternoon, or early evening, the light is always coming from the top. So the highest intensity light is up here. The bottom is always shaded. Which side is warmer? The top. Which side do you have stomata? The bottom. Why? Let's leave the animals out of it for now. Somebody's hand went up and I'm not sure where it was. Oh, yes, sir. You lose less water if you put the stomata down here. Why do you lose less water? Yes, ma'am. You were thinking diffusion, so diffusion goes from high concentration to low concentration. Where do you have high concentration of water vapor? Inside the leaf. Why? Because these cells are 95% water. And so water evaporates from the surface of the cells into the air spaces, and the outside of air, unless if it's raining and it's 100% humidity, the humidity is always less than 100% outside. But inside of the leaf, the humidity could be up to 100%. So the concentration of water vapor is always higher inside the leaf than outside. So diffusion would be down the gradient if there's a passageway. Why do you want to put your passageway down here and not up there? Yes, ma'am. All right, so the temperature will affect the vapor pressure difference, and the vapor pressure difference will affect the diffusion rate. So if you have your stomata up here, you will have a higher diffusion rate because it's warmer. And if you have your stomata on the cooler side, you will have a lower diffusion rate because of the vapor pressure differences. And so you will lose less water if it's on the cooler side. I'm sorry? The temperature will affect the vapor pressure differences, which then affect the diffusion rate. So the warmer temperature at the top means a higher concentration gradient and more water loss. The cooler temperature at the bottom means less water loss because of the differences in vapor pressure. Yes, sir?
Could it also be because if the stomata were on top, they would face less direct sunlight, therefore have less damage. If you have damage to the stomata, you can regulate
So I think the question was about mutations in the stomata or damage to the stomata. So what kind of damage would we be talking about? Direct sunlight can cause mutations because of UVA and UVB radiation, and there is evidence that the cuticle does help protect the cells from those radiation. And the guard cells are covered with cuticle, but when the guard cells open, the hole is not covered with cuticle. So the guard cells are covered with cuticle, so we wouldn't worry necessarily about that. But interesting idea. It could work if they weren't covered, but they are covered with cuticle. You also have to think of the CO2 concentration. So water vapor is not the only thing. The whole point is you want to maximize the CO2 coming in for the water that you're going to lose. Anytime you open the stomata, you're going to lose water. So you have to maximize the gain compared to the loss. So if you think of the demand for CO2, where in the leaf is the demand the highest? On the top, why? Why is that where most of the photosynthesis takes place? All right, high light intensity as well as most of your chloroplasts are here. Tightly packed, palisade mesophyll cells with lots and lots of chloroplasts. High rate of photosynthesis. Down here, less light, loosely packed cells, fewer chloroplasts, lower rate of photosynthesis. So demand for CO2 is less down here, demand for CO2 is highest up there. If you put your stomata at the bottom, when they open, where is the highest CO2 concentration? Outside. And where's the lowest CO2 concentration? Where inside?
This is what I cannot do with the pin.
The lowest CO2 concentration will be here where the demand is highest. So the highest rate of photosynthesis means that CO2 is used up quickly. So the gradient will diffuse the CO2 from the outside through the leaf to the top. Which means all of the cells get a chance to get some CO2. If you reverse that, put the stomata at the top. Well, the highest demand is up here, CO2 comes in, you use it. The bottom may never get any CO2, which wouldn't be ideal. So engineering-wise, it kind of makes sense to have the stomata at the bottom if the light is always on the top surface. So next week we're going to look at vertically oriented leaves and randomly oriented leaves, and then move into details.